Hey guys, welcome back to the channel. So in today's video, we're going to be talking about skeletal muscles specifically. We're going to be talking about its structure, functions, right, and how they are arranged basically, like macroscopically and microscopically, and how contraction occurs and stuff like that. Okay, so basically today we'll focus mainly on its structure and its characteristics and structure. Right, so first let's compare the differences between the three types of muscle cells or muscular cells. So the first type we have, of course, is going to be skeletal. Second type is going to be smooth. And third is cardiac. Right. So skeletal can be found. Where can they found? Location. For skeletal, they can be found attached to bone around the body, right? Attached to bones via tendons. Right. Smooth, normally in organs, or in the wall of blood vessels and basically like organs, basically like soft organs, for blood vessels and basically everywhere else that is not skeletal and cardiac. Cardiac can only be found in the heart, right? Now, what about its function? So its main function is to basically, skeletal, first of all, is allow movement, of course. Its secondary functions, so I'll write one. Secondary functions are basically stabilizing skeleton, allow, so third one, allow certain openings to be open in your body, like major openings. Allow major to be open and closed, so control. Right, so for example, your mouth, if you're opening your mouth, if you're opening your jaw, if you're using your skeletal muscles in your jaw, open your mouth, you can eat food and stuff. Like that's skeletal muscles. Stabilizing skeleton, allow major openings to be open and closed, and monitor the lumen. Stabilizing, protecting inner delicate organs. Protect inner delicate organs, right? For smooth, it basically control diameter of vessels, like blood vessels, for example, and any other type of contraction, basically. The smooth, like for cardiac, basically for heart contraction. And that's it. So the function, those are the function, the nature of their movement. So skeletal is the only voluntary muscle cell type where you're actively controlling the movement and contraction of skeletal muscles. Whereas for smooth and cardiac, they are both involuntary. So you can't control consciously. Like you can't think about increasing the diameter of your blood vessels, and the smooth muscles will do it. No, you can't just do that. It's subconsciously controlled by a bunch of different factors as well, but mostly your subconsciousness, right? And for example, cardiac muscles as well, you can't tell it to stop beating or stop contracting because that's not how it works, right? So this is the skeletal muscles are the only type of voluntary muscle contraction there is, right? Let's say characteristics. All right, so first of all, this guy is multi-nucleated. That means they have many nuclei. For smooth, you only have around one, so, or uni-nucleated. Same as cardiac, uni-nucleated. Right? Skeletal are striated because of the arrangement of proteins of the sarcomeres in your skeletal muscle fiber. So I'll go through that in a moment, but just know that they're striated, or they have stripes along its structure that looks like bands like this. Striated. These guys are not non-striated. Same thing. Maybe cardiac muscles are maybe. I'm not too sure about this one, so this is a question mark. But these cardiac muscle cells can be striated. I forgot which one it is, so I have to double check on this information. And yeah, so these are like the main characteristics, structure, and functions, like a quick summary between the three types of muscle cells. Right, and now let's talk about skeletal muscles basically, overall. Right. Oh yeah, I forgot to tell you guys that for this one as well, functions, there's actually a lot of other ones, like for example, energy and protein storage, right? You're storing your energy in there. And then also when you are cold, you shiver, right? So there's quick burst of muscle contraction or muscle twitching, which produces heat. So it converts chemical energy to heat energy, right? So that's another secondary function, which is not too important, right? So let's go down into its structure, microscopically and macroscopically. So let's try and draw this. Give me a second. Let me try and there you go. Okay. Just like that. So this right here is the biggest structure overall. This is your muscle. Right, muscle. What I'm drawing right now is a fascicle. Same thing, basically. Same thing, okay. This is your muscle fiber. And this will be your myofibril. And let's try and draw. That's tough. There you go. So let's break it down, shall we? This is a muscle, right? And a muscle is made up of a bunch of little bundles called fascicles. Fascicles are made up of a bunch of little muscle fibers. So muscle fibers are basically muscle cells. They're the same thing, right? But we don't call them cells, muscle cells. We call them muscle fibers because of its long structure, right? So its diameter can be a few micrometer, but its length can go up to a few centimeters. So it's very long. It's uninucleated, it's striated. You can see these type of bands or line going down here. And muscle fibers are made up of a bunch of bundles of what we call myofibrils. And myofibrils are made up of what we call these repeating unit, contractile repeating unit, or called sarcomeres. Sarcomeres are basically this. Let me draw it out for you. This is what a sarcomere looks like. Oops, this shit drawing. Here we go. This in the middle right here is the thick filament, which is the myosin. This is the thin filament, which is the actin, right? So the way I remember it is actin for thin. There's this line down the middle called the M line, right? There's this right here band, we call this the H band. So that this region where there's only myosin, there's no actin as well. If you want the bigger version, then you've got this one right here, which is called the A band. It goes from both ends of myosin, which includes a little bit of actin as well. And the rest of the sarcomere is called the I band. Right, same as this side as well. The rest is called the I band on the side. M line goes down the middle. This is the Z line, right? The zigzag line where all the actin filaments connect together. It looks like a Z, that's why it's called the Z line. M line's down the middle. H band is the part where there's only myosin, so no actin. And A band is the part where there's both actin and myosin. So it goes from both ends of myosin, which also contains a little bit of actin on both sides. So this is a simple structure of a sarcomere. And sarcomeres together make up myofibrils. Myofibrils are found, there are tons of bundles of myofibrils inside a muscle fiber, which have a lot of nuclei, lots of mitochondria, this sarcoplasmic reticulum. So we'll go through that in a moment, but this is the basic structure of muscle, right? So you've got these white dots here. I mean, they're not white, but these small dots here, these are actually your blood vessels, right? So muscles are highly vascular. These are your blood vessels. And these are your fascicles, right? The layer outside here is called the epimysium, or mysium, I forgot which one it is, but the outermost layer, so this layer right here, the one I was shading right now, this layer is the epimysium. This layer in the middle the tissue in the middle between the fascicles and the blood vessels are called the perimysium, right? And then in the tissue between the muscle fibers in a fascicle is called an endomysium. And you just have to know that these types of tissues are connective tissues that ensheath around. So endomysium is a connective tissue layer that ensheath around each individual muscle fiber in a fascicle. You have to know that perimysium is a connective tissue layer that ensheath around each individual fascicles in a muscle, and epimysium is the connective tissue layer that ensheath the entire muscle itself, right? So that's the basic structure of skeletal muscles, breakdown of fibers as well. And then also these are blood vessels, but you can also find nerves, neurons inside muscles as well, because of course contractions are controlled by neural activity or your brain activity sending signals down to your muscles. So let's move on. What happens is basically once your brain sends signals down to your muscle fibers or your muscle cells, a chain of reaction will occur basically, and muscle fibers will contract, and we will cover that in the next lesson. So let's talk about, so as you know, skeletal muscles are attached to bones via tendons, and tendons are a type of dense fibrous connective tissues. So many bands of tissues come together to form tendons. And they join the skeletal muscles to the bones or the skeleton. Right. Now let's draw what a muscle fiber would look like internally. Right. So let's kind of visualize what it would look like. Okay, start from there. Just like that. Let's try and imagine what they would look like. Just like that. This is what it would look like. So one, two, three, four. You've got these guys continuing on. Just like that. Okay. So this right here is the sarcolemma, or the cell membrane of a muscle fiber, right? Again, a muscle fiber is a muscle cell. Sarcolemma is the same as the cell membrane of a cell, right? These are sarcolemma. This right here is a myofibril. I'll just draw these little dots in here, right? These tubular-like extensions, right, that goes along the myofibrils. You've got this part as well. We call them the sarcoplasmic reticulum. Sarcoplasmic reticulum. Same as your endoplasmic reticulum in normal cells. Spaces between them is called the sarcoplasm or cytoplasm in normal cells. But then the difference is that you've got these invaginations or extensions of your sarcolemma running down deep into from superficially on the sarcolemma deep into the muscle fiber. We call these the transverse tubules or the T tubules, which are extensions of the sarcolemma or invaginations, which will carry action potentials from the surface deep into the muscle fiber to reach all of the myofibrils and allow contraction to occur, right? And that is the basic structure of also these here. So this bit in the middle is a T-tubule. These two bits at the end here, this band right here is called the terminal cisternae. Terminal cisternae. Cisternae of the sarcoplasmic reticulum. So basically the ending parts of the sarcoplasmic reticulum. And these three together, the T tubules in the middle and the two terminal cisternae or cisternae of the sarcoplasmic reticulum on both sides together are called the triad. And this is where they interact with each other, right? And the action potential is sent across here. And this is basically the structure of what it is. Okay, so you also need to know that sarcoplasmic reticulum main function is to store Ca2 plus ions, which are very important in muscle contractions. You do need these. And yeah, these are basically the structure of a muscle fiber. Very briefly, all right. But then after that, we also need to know about neuromuscular junction. Oh, it's quite simple. It's an introduction basically, but you just need to know that a neuromuscular junction, neuro, Neuro, muscular junction, or abbreviated NMJ, is basically where you've got your you've got your neuron interacting with a special place in your sarcolemma. So this is your sarcolemma, and this is your presynaptic neuron, which is the motor neuron. And this special bit right here, where this special kind of like increasing surface area bit is called the neuromuscular junction. So this space right here, the neuromuscular junction. And you also need to know that a muscle fiber receives contact from one neuron at one site, and this is one only, right? But a neuron can innervate many different muscle fibers at the same time, right? So imagine you have this one neuron, right? One motor neuron right here. You've got your axon terminal bonded to, you've got like, for example, one muscle fiber here. You've got another one running here. You've got another one, and you've got another one. So for example, 4 of them, right? So this is what you call a motor unit. A motor unit is basically the motor neuron and all the muscle fibers that it innervates. But so a motor neuron can innervate many muscle fibers, but a muscle fiber can only receive contact from only one motor neuron only at a specific site called the neuromuscular junction. And that's all about it. You also need to know about excitation contraction coupling. So what happens is that you've got this bit right here. This is your T-tubule, as you guys know. Let's imagine this to be your sarcoplasmic reticulum. Right. Here we go. Right, so action potential travels along the sarcolemma and down into the T-tubules, like that. As the action potential reaches the T-tubule, the DHPR, voltage-gated protein channel, will be activated. Right, so once the action potential reaches the T-tubules, the DHPR will be activated, allowing calcium ions, Ca2+, will flow through. This right here is the ryanodine receptor, abbreviated R, little y, capital R, will detect the Ca2+ that has been let through. So basically the RYR receptor, the ryanodine receptor, will interact with the DHPR to basically release even more calcium ions into the sarcoplasm, right? So this white bit over here is the sarcoplasm. This bit over here is just the synapse. Basically, there's nothing in between here. This bit is empty space in your cell, or your extracellular matrix. You can say that, right? And then so once DHPR voltage-gated protein channels are activated and Ca2+ ions are let through, right? So what happens is that the receptor over here will detect the Ca2+, will open itself up and allow even more calcium ions from the sarcoplasmic reticulum to exit out and bind onto stuff on the actin. And so basically the calcium ions will allow muscle contraction to happen. And that's the extent you need to know for now. And once the stimuli has run out, once all of them, or when stimulation ends, or when your muscles feel fatigue, what happens is that the calcium ions are not needed anymore. So they're recycled by the sarcoendoplasmic reticulum calcium ATPase, abbreviated SERCA, circa, active. So it will actively pump, this is a protein pump / channel, will actively pump Ca2 plus ions back into the sarcoplasmic reticulum to recycle them to be stored again after muscle contractions are over. All right. And that is basically all you need to know for today. And we'll see you guys in the next video when we cover more in depth of the electrical signal as well as other factors affecting muscle contraction. Thank you, and we'll see you then.